All right. So I want to first of all give thanks to everybody that is online right now and is attending this presentation, taking some of their time to come here. What we have to say and what this project is about. And I'm Placido. I work in Dr. Brambilla's lab. And one of the projects that I've been working on lately is about the TNFR2 signaling and how to enhance it in the CNS in the context of neuroinflammation. As many of you know, Brambilla Lab studies multiple sclerosis, which is a T-cell mediated autoimmune demyelinating disorder. And in multiple sclerosis, what's observed, especially in the central nervous system, is acute inflammation, and that can be one of the causes of oligodendrocyte cell death, alongside with the autoimmune activation. Oligodendrocyte cell death can be one of the causes for demyelination, alongside with the inflammation. And ultimately, what we observe is the appearance of axonal injury, neuronal loss, which can be the cause of an impairment of the CNS functions that can be most of the times irreversible, especially in some of the stages of the multiple sclerosis pathology. What we study in our lab specifically is the signaling of TNF. What is TNF? TNF, maybe most of you know about this cytokine, tumor necrosis factor, and is multifunctional and regulates a lot of different processes. It is studied in the context of inflammation, autoimmunity, in cancers, but also in infection diseases. And TNF signals through two different forms. The first form of TNF is transmembrane TNF, which binds preferentially to TNF receptor type 2, that I'm gonna call from now on TNFR2. And it has a higher affinity for this receptor, and it initiates a series of cascade signaling that brings to pro-survival effects alongside resolution of inflammation, myelination, and remyelination in the context of neuroinflammation, as we're interested into, but also the maintenance of immunity to pathogens. TNF, as shown here by the arrow, can be cleaved by TACE enzyme into its soluble version, which is soluble TNF. Soluble TNF binds preferentially TNF receptor type 1, which I'm going to call TNFR1 from now. And the activation of this receptor leads to both pro-apoptotic and pro-survival signals. This is being observed in many kinds of diseases, and toward the shift towards one or the other depends on the context where TNFR1 signaling is studied. And also, activation of TNFR1 and its pathways is associated with chronic inflammation. So now, why do we study TNF in the context of multiple sclerosis? Many years ago, and we're talking about probably the end of the 80s, the first associative studies that scientists carried out were between the levels of TNF in MS patients in the serum and the cerebrospinal fluid compared to age-matched unaffected individuals. And in this seminal paper from the New England Journal of Medicine of 1991, we can see that the disease severity, so you can see here as EDSS score, increases as the TNF concentration in serum also increases. And the same is true for the progression index of the disease of multiple sclerosis, alongside with increasing the TNF concentration. So there was this association between increase of TNF and increase of disability score and progression. Also, post-mortem studies allowed to study the active lesions that were present in the spinal cord or in the brain of MS patients. And what was observed was that the levels of TNF were elevated in these periods. Furthermore, TNF levels correlate again with the lesion and disease progression. So just to reiterate this, which is very important. Now, in the context of multiple sclerosis, we were talking about the late 80s and a lot of research has been carried out. But what was most important was the establishment of a model for multiple sclerosis, specifically an animal model of MS. And this model, which is the one that we also use in our lab, it's not the only one, obviously, but it's the one that is most recognized and used, which is the experimental autoimmune encephalomyelitis, also acronym as EAE. In EAE, most of the times, this is what I tell my students, and take it with a grain of salt, it's a little bit of a stretch, but what I say to make students understand, I say that we make mice allergic to their own myelin. Now, the word allergic also appeared in the early definition of EAE, and in the first place, the A stood for allergic. So practically, what we do, we stimulate the immune system of these mice, and then we immunize them with specific antigens that are myelin specific, and there are different kinds of antigens that can be used. Obviously, there are different protocols for that. And the concurrent BBB breakdown, brain-blood barrier breakdown, and the immune response towards this antigen can initiate the infiltration of peripheral immune cells within the central nervous system. Now, when this happens, mice do not show immediately disability, which is a flaccid-like paralysis. But after a certain amount of time, they start to display this phenotype, which can be scored in a scale that, in our case, ranges from 0 to 6. But obviously, there are different ways of grading it. The most important take-home message from the scoring of the flaccidity in this mice is that it goes from bottom up. So it goes from the tail, with a loss of tail tone, then flaccidity, and then it can eventually extend to the hind limbs and forelimbs. Now, this is what the model has been used in many studies and also we use in our lab with a specific induction protocol. In many years, the studies that have been carried out on TNF and the EAE animal model brought to this. Obviously, this is a quick summary, where the ablation complete of TNF or TNFR2 germline knockout led to exacerbation of the EAE pathology, as well as impaired remyelination, while TNFR1 knockout mice were completely resistant to EAE. So from these studies, we can briefly, obviously there is a lot of literature supporting this. We can say that TNFR1 signaling through soluble TNF is detrimental in the context of neuroinflammation EAE, while TNFR2 signaling, which is activated by transmembrane TNF, is actually protective. And this is what we actually focus in our lab, and majority of the projects focus on the TNFR2 signaling and its protective role. Now, the first paper that our lab published back in 2011 focused the attention on the TNFR2 in the context of EAE as a model of multiple sclerosis. You can see here, on the ICD arrow, that TNFR2 is expressed throughout the glia in either astrocytes, oligodendrocytes, throughout maturation of oligodendrocytes, so adult and oligodendrocyte progenitors, as well as microglia. To our knowledge, TNFR2 is not expressed in neurons. In this study, mice were induced with EAE, and here you see an EAE plot. I will spend a little bit of time on this EAE plot, and then I'm going to go into the pharmacological treatments that were done in this study. We have on the x-axis the days after the immunization, so time practically, and on the y-axis we have the clinical score. So mice are immunized, so they are induced on day 0, and they do not show symptomatology for around 10 to 12 days, obviously with some variability at the onset. Now, after the onset, we observe a surge in the disability score, which means an increase, progressive increase, day by day of the paralysis. Again, bottom up. And then after this acute wave, mice partially recover. Now, here we want to stress the pharmacological treatments that were performed in the studies. Now, the first group is vehicle in a black square, and then mice also were treated. A group of mice was treated with etanercept, which is the empty circle. Etanercept is an anti-soluble and anti-TNF as a decoy for complete TNF ablation, and therefore there is no activation. In the context of the EAE, what happens is that with this treatment is that after the initial wave of acute inflammation and acute disability, mice stabilize, and then at a chronic stage, we observe a further increase in the mean score, clinical score, which stays constant up until 55 days after induction. This kind of treatment was also reproducing data that were observed with the administration of a lenacept, which is very close related to etanercept, that was actually done in a clinical study back in 1999. So, which was suspended because the rate of relapses and the increase of the number of relapses was so high that the study needed to be stopped. Now, the other group that we observed here in this experiment is the treatment with Expro 1595. These mice are treated, and then on day 16, and then after a stabilization time, they start to recover, and their recovery stays constant, especially as we can see at later time points at the chronic stage. So at this chronic stage, the anti-soluble TNF Expro 1595 exert a beneficial effect in the EAE. And this group, the mean score, is statistically significant and lower than the vehicle group and obviously of the etanercept group. In the paper, all the details about how this happens and what regulates obviously are described. But for the sake of this presentation, I'm showing you this part where the pharmacological inhibition of TNFR1 is protective in the context of the EAE. Now, for us, just 1 second, I had to exit. I don't know what time. It happened before as well. My bad. Okay. In our studies, we want to assess the role of TNFR2 in the different glial cell types. And in order for us to do this, we make use of Cre-LOX systems, where we have our transgenic mouse that has the TNFR2 gene fluxed by 2 LOX-P sites. And in the presence of expression of a Cre recombinase, also known as molecular scissors, the LOX-P sites can be flipped out and then the gene excised, therefore bringing to a conditional ablation. Now, in terms of the other mouse that is needed for obtaining a double transgenic where this system works, in this case, we use several kinds of Cre specific promoters. So where the Cre-ER gene is under the control of a cell-specific promoter, and therefore the expression of Cre-ER is restricted to this cell type, depending on the promoter that is chosen. Now, the presence of the ER, which is an estrogen receptor on the Cre, determines the activation state of Cre recombinase. So what the ultimate thing is that the treatment with a specific drug, tamoxifen, that binds to the ER leads to the Cre activation and thus bring to the gene ablation. The administration of tamoxifen can be performed with different protocols, but can be chosen and can be time controlled. And this allows us, this overall system, to ablate the TNFR2 from specific cell type and timed. So we, in different studies, use adult mice because we do not want the ablation of TNFR2 occurring during development, either at embryonic stage or after birth. And we have virtually all kinds of mice for the ablation in glial cell types. Now, in this study that he's in preparation and is project that Aritha, our grad student in our lab, is developing, we studied the TNFR2 ablation in mature oligodendrocytes. And the first evidence is that the ablation of TNFR2 in this specific population of oligodendrocytes is detrimental. In order to do this, we use PFP-CREAR mice, where the ER cree is activated upon tamoxifen administration and ablates TNFR2 gene specifically from these mature myelinating oligodendrocytes. And here we can see the EAE plot, which is similar to the other, where after a certain time of absence of disability, our double transgenic mice initiate the display, show the symptomatology at earlier time point and a higher score, and this occurs in the early phase, so in the acute phase. Obviously, this data, I want to thank Arita for sharing. These data are still not partial, but still preliminary, and we're working on this project. But this is for us to see that the ablation of TNFR2 in the context of the EAE from oligodendrocytes that are actually myelinating is detrimental. So my whole point as a first conclusion is that the pharmacological inhibition of the soluble TNFR1, soluble TNF, TNFR1 signaling is protected in the EAE context, as well as the ablation of TNFR2. We observe, the ablation of TNFR2 in mature oligodendrocytes instead is detrimental in the EAE context. Now, this brings us to the hypothesis, our overarching hypothesis, that the possible potentiation of TNFR2 expression in this specific cell type, which is mature oligodendrocytes, can be protected in EAE. Now, in order for us to prove our hypothesis, the aim was to, first of all, be able to generate mice that are transgenic that can overexpress TNFR2, and then these mice had to overexpress TNFR2 specifically in mature oligodendrocytes, and then on a third stage, study this overexpressor during the EAE in the context of neuroinflammation. Now, moving on to aim number one, the overexpression of TNFR2 was achieved thanks to the generation in the lab of a specific construct. This was done in collaboration with Morai's lab, and specifically with Nadi, as you can see here. And she generated this construct where the TNFR2 gene and a reporter gene are under the control of a specific TRE sequence, which is TRE stands for tetracycline responsive element. Now, this is a bidirectional system. Then when it's activated, initiates the expression of TNFR2 and EGFP at the same time. Therefore, TNFR2 is overexpressed in a system that already expresses basal level of TNFR2, and EGFP, as non-endogenous, is expressed and can be used as an index of overexpression. Now, these, you can see here, are HeLa cells that were transfected in the TNFR2 vector, and 24 hours and 48 hours specifically after the induction, these cells show fluorescence for the GFP. So this was the proof that a construct was properly done. Obviously, the details, molecular details, are much more complicated than what I'm showing you, and that the construct was working. Now, we took this construct and we sent it to Cyogen for the generation of a transgenic mouse. So for the integration of this transgene within the genome. And we obtained back from Cyogen 4 founders, A, B, C, and D. You can see here the PCR, which shows the presence of the transgene, and also compared to the GFP control, which was the construct that was generated in the first place. So these 4 mice, 4 founders were viable, survived, and were transgenic. We then move forward, and we actually had to make sure that these founders were actually work, the transgene were actually work. So we had a problem of founder selection. In order to make TRE system work, this TRE system, tetracycline responsive element system, need to be crossed with another transgenic mouse, which is known as transgenic TTA or eventually RTTA system. A TTA system, which is a tetracycline transactivator system, those are more recent and more accurate. You will understand why in a few. In these systems, the TTA gene expression is under the control of a cell-specific promoter. So it's the same thing as I showed you before in the Krylov systems. The selection of this promoter allows for the cell specificity. Now, what is that is expressed? In this case, we have this tetracycline transactivator. Now, in the presence of doxycycline, these systems are suppressed. Why? What happens? In a double transgenic mouse that carries both transgenes, which is the TTA transgene, regardless of the cell specificity, and the TRE transgene, the presence of doxycycline blocks the tetracycline transactivator from binding to the TRE. When doxycycline is removed, TTA is free to bind to the responsive elements and therefore initiate, in this case, the double expression, overexpression of TNFR2 and expression of the GFP protein. The doxycycline is commonly used in the chow, so in the food, and the removal of the food can be done in a very easy and non-invasive way at any time point and allows for the time-controlled start of the overexpression. This is a concept that's very important because we're going to go back to this several times during this presentation. Now, our transgenic mouse, the TRE TNFR2 that bears concurrent TNFR2 and EGFP overexpression, was crossed with a specific TTA transgenic mouse, which was CAMK2 alpha, under the control of CAMK2 alpha neuronal promoter. This transgenic mouse allows for the overexpression in the neurons of the TTA, taking advantage of CAMK2 alpha, which is a very strong neuronal promoter. Why did we choose CAMK2 alpha TTA? For, again, the aim was to select the founders. Because TNFR2, as I told you at the beginning of this presentation, is non-neuronal. And in this case, when given the fact that TNFR2 is practically absent in neurons, even a minimal TNFR2 overexpression could be detected. And also at the same time, we're choosing a promoter which has a self-specificity for neurons within the CNS that are obviously highly expressed in the CNS. And it's also a very strong promoter. So we took advantage of this whole system to select our founders. This is the protocol that we used. I hope it's clear to understand. So on here in purple, we observe the breeding between the two transgenic occurring under doxycycline treatment. Simply, mice are fed doxy food. Now, after the breeding, mice are born, and the offspring is kept on doxycycline until 21 days. And this is when mice are usually weaned off from the parents, as well as, in this case, weaned off doxycycline. The weaning of doxycycline allows for the TTA to be active and bind to the TRE element, and again, express the two proteins of interest. Mice are kept in this condition up until 90 days, where tissue is collected for molecular analysis. And this is what happened. For our different 4 founders, we had line A and C, which did not work. Practically, we had a double expressor, so double transgenic, but for some reason, there was no overexpression of, in this case, GFP protein of interest. This could be for many reasons. One can hypothesize because the location of this transgene in both cases is in some parts of the genome where it's not possible for the TTA to bind to the TRE elements. There are many possibilities, but fortunately, we had two lines where the overexpression actually occurred, which were line B and line D, where the overexpression in a line D of the GFP was higher. So we wanted to look into the line D better, because although the system has this double initiation of the expression concurrent of GFP and TNFR2, until we don't really see that TNFR2 is really overexpressed, we can't say that the transgenic system is working. Therefore, for line D, we focused on the overexpression of TNFR2, and you can see here the band at 70 kilodalton that is stronger for double transgenic mice compared to wild-type litter mates. Therefore, we have both. We have expression of GFP and overexpression of TNFR2. Yes, I see there are some questions which I will probably address at the end. The overexpression of TNFR2 also needed to be proved. We had to prove that was not leaky. And what do we intend as leakiness? The leakiness is a specific phenomenon that occurs when there is an ectopic expression during suppressive condition. So in order for us to assess there was no leakiness, we had to assess there was no expression of EGFP nor TNFR2 during doxy treatment. Therefore, here we use the same paradigm as before, but we kept mice up until adulthood on doxycycline food. In this case, what we observed for the line D, which was the one that we selected, no expression of GFP again, and lower expression of TNFR2, which is minimal, and it is detected here, and it's lower than the mice that were kept off doxy. So it goes back to baseline levels, and at this point, this was the confirmation that our transgenic model works and it's not leaky. Further, we had to assess that the crossing with the CAMK2 alpha TTA was allowing the overexpression of these two proteins of interest in the cell type that was wanted. In this case, were neurons. You can see on the first strip the colocalization of EGFP positive and NUN positive cells in the cortex layers, as well as the overexpression of TNFR2 in the neurons on the lower strip, specifically occurring at the membrane site, being the TNFR2 and membrane receptor. The location of the expression of TNFR2 was also demonstrated to be correct. So at this point, we can conclude from this part that we were able to obtain an overexpression of TNFR2 thanks to the generation of a transgenic TRE-TNFR2 construct. We also had a time control of the overexpression through doxycycline removal, as well as the cell-specific overexpression in CAMK2 alpha positive cells, which are neurons. Now, we were able to move on to the second phase of the project, which was to obtain overexpression of TNFR2 in mature oligodendrocytes. In order to do this, I showed you here a little bit of the maturation stages of oligodendrocytes that ranges from the oligodendrocyte precursor cells, OPCs, to the pre-oligodendrocytes and to the mature myelinating oligodendrocytes. Here you can see different genes and that are used as markers for the different stages of oligodendrocyte maturation. And we see that the PLP, which is the last one, is specifically localized at later stages when the oligodendrocytes are mature and myelinating. Therefore, we were able to use PLP control the TTA expression, and in using the PLP TTA transgenic mice under doxy condition, so that the system will be suppressed where mice were crossed again with the TRE TNFR2 transgenics, and the offspring that was double positive, that was born in doxycyclic condition, would not allow for their overexpression of the protein of interest until doxycycline was removed, TTA binds TRE, and again, we have the expression of TNFR2 GFP. Now, you can see here the very widespread localization of the EGFP throughout the brain and the spinal cord in the areas that we expected to happen, either in the cortex, in the corpus callosum, in the areas of the striatum, as well as white matter and gray matter of the spinal cord. But the expression of TNFR2 alone doesn't allow us to say that this was a cell specific. Therefore, we counterstained the slides for OLIG2, which is a marker for oligodendrocyte cell lineage, as I showed you a couple of slides ago, as well as for APC-CC1, where we observe co-expression, and then we observe that then we have the overexpression in mature oligodendrocytes. Now, this overexpression does not occur in the other glial cell type, neither astrocytes nor microglia, and also does not occur in neurons. So we were able, using our TRE transgenic with the PLP-TTA, to induce the overexpression of our transgenic system. So we were finally able to tackle our last and most important, our main focus, which was the study of the TNFR2 overexpressor in mature oligodendrocytes in the context of neuroinflammation and EAE. In order for us to do so, I'm proposing you again the same very close paradigm that we chose for the CAMK2 alpha mice. We bred PLP-TTA and TRE transgenic, kept them on doxycycline, offspring was born and kept on doxycycline until weaning, and then from weaning up until 90 days, so up until full adulthood, the system was withdrawn from doxycycline, simply we changed the food, and the overexpression was allowed to occur in non-neuroinflammatory conditions. And then when a fully mature mouse, they were ready, they were induced with the EAE and kept from for a chronic time points from 40 to 45 days after induction, and then sacrificed for molecular analysis. Now, we did this study in both males and females. And I'm going to show you from now on data regarding the two different sexes, and we're going to see some differences. Now, in male mice, this paradigm, the early dox, because doxycycline is removed at weaning age, does not affect the rotarod performance, so therefore does not affect the motor phenotype, not during the training and not at test. This is our protocol. That is a 7 days protocol for rotarod. But so this allows us to say that the activation of the transgene in male mice does not affect the motor phenotype, and therefore we're able to induce neuroinflammation and to score the disability score on a mouse that in normal condition has a normal motor phenotype. But what we found with this paradigm in male mice, that the early dox paradigm does not affect the EAE outcome. It does not worsen nor ameliorate the outcome of EAE. And this is possible to see in this EAE plot again, where we observe the x-axis, the time, and then the score, which increases after 8 to 12 days. We have occurrence, and then we get a chronic stages where there is practically no statistical difference and significance between the two groups. Although there was some indication with, obviously we have enough power to say that those are not statistically different. There was some indication that maybe that could have been something protective happening, at least from a very broad perspective of mean clinical score, in the double transgenic, which obviously was not the case using this paradigm. And we thought that the early dox was actually too early for removal of doxycycline. And we thought this for several reasons. One of those, which is the most important, is the fact that this same paradigm used in female mice leads to motor impairment in non-inflammatory, neuroinflammatory conditions. So naive female mice, after overexpressing TNFR2 or after the overexpression activation of transgene, at adult stage, they show lower performance and motor impairment at the rotor rod, either in pre-training or in test condition. And second, they actually display a worsening of the EAE outcome. And this was present at the early time point, which is the acute time point. Now, at this point, many doubts were raised, and I have to be honest, I was very puzzled, because we were trying to overexpress a protective factor, and in this case, we obtained actually the opposite effect. This, I learned that can happen in research and opens a whole new field of questions and investigations that need to be done. Going into the simplest thing that I could do was to go and read some literature. I could see some studies where the specificity of PLP promoter was studied at the early postnatal and late postnatal stages. In this paper from 2010, the authors assessed that the PLP promoter is highly specific for mature myelinating oligodendrocytes only after P28, therefore like a month-old mice. Now, if this is the case, we can hypothesize that in our case for females and also for males, but to a greater extent in females, we might have a lack of specificity of the overexpression, which can interfere with the developmental myelination. Always in this study, it was proven that the myelination occurs during embryonic stages, of course, but also in postnatal life up until P28. And PLP promoter is not specific for mature oligodendrocytes before that milestone age is reached. So this could be one of the explanations why we observed a worsening EAE outcome and worsened motor impairment in the non-inflammatory conditions for females, and practically no protective effect for males. So no protection and actually a bad situation for females. So we had to go back to our paradigm because of this paper and change the time where the withdrawal of doxycycline was performed. So this was what I showed you before, the early dox paradigm, and we shifted everything rightwards into a late dox paradigm where mice after weaning off their parents are still kept on doxy food up until P75. And then they are allowed for 2 weeks from 75 to 90 of the overexpression of the system in non-inflammatory conditions that we can call naive. And then after, EAE was induced at 90 days of age. Now, again, in order for us to assess disability score, we had to make sure that in naive condition, mice were not impaired. And for male mice, this was true, where we observed no impairment at the rotor rod, either in pre-training and in test conditions. When we induce at 90 days mice with the EAE, lo and behold, we have the EAE outcome, and we observe with this paradigm a reduction of the mean clinical score compared to the control group in double transgenic, starting from day 20 up until chronic status. So in this case, the overexpression of TNFR2 in mature oligodendrocytes for male mice is protective at chronic stages of EAE. I remind you that in the early dox paradigm, there was nothing happening. And there was a tendency to see what we actually see here, but it was not statistically significant. So we wanted to go ahead and characterize better what happens in these mice at the chronic stages. So we started to address better at molecular level what are the things that are occurring during these stages of EAE. And the first thing that we did was the staining for myelin, which is black gold staining. Black gold staining is a specific staining that we performed on the spinal cord section from chronic EAE mice. And it is possible to see the demyelinated areas, especially on the borders and in different areas at this point in the chronic EAE. The demyelination occurring can occur also patched. And in our control mice, we observed a higher white matter lesion. So there was a higher percentage of white matter that was damaged during this time compared to our double expressors that overexpress TNFR2 in the mature oligodendrocytes. Then we moved further and we wanted to look at not only demyelination, which obviously is the first thing I wanted to look at because we had to assess the population of the glia that actually produces myelin. So this was the first thing that was also the faster and the easiest thing to do, was to assess demyelination. Also, we stained for GFAP, and this is still preliminary data that I still would like to show you because they show practically the same thing, but using another cell marker, which is GFAP for astrocytes. And doing a 3D reconstruction of the lesion areas, it is possible to see in 3D the extent of damage that occurred in controlled mice and how much higher it is compared to our overexpressor mice. So we move forward and we move out from the oligodendrocyte cell type myelination, so from the mature myelinating oligodendrocytes onto another glial cell type, which in this case is microglia. Using the flow cytometry and performing it at the chronic EAE, we observed a reduction of the microglial activation in double transgenic mice at chronic EAE. In these mice, we can see either in the spinal cord, but also in the brain, in the cerebellum, we observe a drastic decrease of cells that are, of microglial cells that are activated. And on the right hand, you can see the flow cytometry plot where we first selected the CD45 low CD11B positive microglia from the spinal cord and cerebellum, and then gated them on MHC2, which is a marker for activation. And these are examples of how much higher is the MHC2 percentage of positive population for microglia in the controls compared to our double transgenic. And this is true in both spinal cord and brain. In my little experience, it was stunning to see that the reduction of microglial activation could occur in spinal cord, but also in brain. Spinal cord is the one tissue, CNS tissue, that is mostly affected by EAE. And obviously, the immune cell infiltration occurs also in the brain, but to a much lower extent than what happens in spinal cord. So this came to be as a very good surprise that the protection induced by the TNFR2 expression in myelinating oligodendrocytes not only prevented demyelination, but also reduced the microglial activation in both CNS brain and CNS spinal cord. Now, the flow cytometry data that we obtained also range on the more general immune cell infiltration. What we do with flow cytometry, we go ahead and we stain markers, using specific markers for antigens that are expressed by T cells, T helper cells, B cells, so immune cells that in general, this is just to say, do not belong, these immune cells do not belong to the CNS parenchyma because those are infiltrated after neuroimmune, neuroinflammation, EAE. So we're in a chronic stages. And the pattern of immune cell infiltration at this point in chronic stage can be different than early stages or presymptomatic stages. Nevertheless, what I'm showing here, what you can observe is a strong significant reduction in the T helper CD4 positive cells, in the T cells cytotoxic CD8, B cells, monocytes that are CD45 high, CD11B positive, and natural killer cells. Also, we observe a reduction of the activation of the B cells and of the monocytes in double transgenic mouse in the spinal cord. So this overall allows us to say that there is a reduced immune cell infiltration at chronic EAE in TNFR2 overexpressors, and also a lower activation of these cells when they are within the CNS. Also, we went ahead and we wanted to observe whether in the brain also we saw some effects, as we saw a lower microglial activation. And in this case, what we saw was a significantly reduced infiltration of B cells in the cerebellum as well. Sorry, I have, again, a problem here with the presentation that fades the background for some reason. 1 second. Okay, back to where we were. And we observed in both B220 and activated B220 reduction of cell number within the cerebellum, so within the brain. And this is also very striking and very important result, that these overexpression of TNFR2 can also reduce and protect the brain from the immune cell invasion in the chronic stages of EAE, because this is when we have done these experiments. And now last, we also analyzed at the peripheral level, the spleen. And we do observe a higher number of the different immune cells in the spleen. This is true for T helpers, T cells, B cells, and also activated B cells and natural killer as well. Now, we believe that the induction of the EAE, which obviously generates an immune response, in this case, is confined to the peripheral system. And therefore, the fact that those immune cells are not able to invade the CNS at either spinal cord and brain level allows us to remain in circulation and obviously to be confined as well as in the spleen. And this is what we think we observe in this case, where the overexpressors show an increase of all the markers that we have across the board. Now, we have overall seen that there is actually a protective effect of the of this overexpression. Wait 1 second. Presentation is stuck. I am so sorry. I don't know exactly what's happening, but all right. So, okay, let's see whether I can move on. Okay. In the last few minutes, I wanted to assess what happens with the late dox paradigm in female mice. In this case, this paradigm did not affect the motor behavior in female mice. So this is a proof, one proof of the fact that the overexpression of TNFR2 initiated in an adult system does not alter developmental myelination, and therefore does not lead to an impaired motor phenotype. Obviously, this is a conclusion that we can draw that would need further investigation. But when these mice that are baseline, naive, normal, we induce them with the EAE, what happens is that we don't see any difference between the two groups, transgenic and normal mice, transgenic and controlled mice, sorry. So the EAE outcome is not affected. But also in this case, what we saw in a different situation for the males, we see a tendency, which is also in this case not statistically significant, to reduction of the mean score at chronic stages. Now, since this system, the altering the paradigm, shifting the paradigm worked for males before, we decided to further change the paradigm for female mice. And we moved from a late dox paradigm, which I showed you before, to a later EAE paradigm, where the doxycycline was also always removed at 2 months old, 2 months of age, but the EAE was induced 4 weeks after mice were weaned off. So for 4 weeks, the overexpressing system would be functioning in non-inflammatory, neuroinflammatory conditions. And also in this case, obviously, we had to assess the motor phenotype before the EAE induction. And what we observed in this case, that also this paradigm, just like the late dox paradigm, does not affect the motor phenotype in female mice. And distanciating further from the developmental stages, first of all, helps with not interfering with that process. When we induced these mice with the EAE, lo and behold, we saw an amelioration or an improvement of the EAE outcome, but not at the expected time point, which was later stages from the data in the females themselves, but also that we observed in the males. But we observed the amelioration of the EAE symptomatology at early stages, so in the acute phase. And the study in the acute phase obviously is different because there is a first, this is the period of time where there is a first wave of immune cell infiltration within the CNS. And therefore, the analysis that we need to run, because this is where I'm standing right now with my female mice data, has to be slightly different than what we studied for mice. So now, in a future work perspective, and with this I conclude, is that in female mice, we have to characterize the EAE phenotype using this specific paradigm, while for male mice, in the late dox paradigm, there are many things that I would like to assess. First of all, is the brain-blood barrier integrity, because if you have peripheral immune cell activations that are there but do not invade within the CNS in both spinal cord and brain, it could be that the overexpression of TNFR2 in mature oligodendrocytes not only reduces demyelination, but also affects the BBB integrity. It is very important and pivotal for us to know also the survival of the mature myelinating oligodendrocytes in these stages of EAE, as well as neuronal survival. I would like also to investigate on the aspects of the activation of the astrocytes as well, since we see a reduction of the microglial activation in both spinal cord and cerebellum. And then we're also planning to investigate the actual mechanism that is driven by the TNFR2 enhancement of TNFR2 activation in the oligodendrocytes. And one hypothesis, and open to suggestions, could be the activation of the NF-kappa B signaling pathways. TNFR2 presides at both, alongside with the TNFR1, NF-kappa B canonical and non-canonical signaling. So it is possible that the overexpression of TNFR2 potentiates the NF-kappa B signaling, bringing to a protective effect in neuroinflammation. So, as a conclusive remarks, we were able to establish a TNFR2 overexpression model, and then we were able to say that the TNFR2 overexpression in mature oligodendrocytes is protective in the EAE context. So now, last, I just want to thank briefly my lab, in the person of my boss Roberta, Arita, and Melanie, our wonderful grad students have been so helpful and so collaborative in this project. My undergrad Shweta, that is now graduating and leaving us. Our technicians Eva and Shafayet, which are amazing and so important for all this the care about the transgenic mouse and the colonies, and as well as Maureen, our newly acquired grad student. I want to thank Morai's lab in the person of Carlos and Nadine, the Miami Project DVR, which has done a terrific job in helping us keeping the animal colonies, and as well as the McKnight Vision Center in Jennifer Arcuri that at the time used to still work there. Now, this project was funded by the Italian MS Society. You can see my boss and I at the presentation in Rome, the meeting that we have every summer. And I want to thank the Italian MS Society for having given me the chance to carry out, as a young investigator, this investigation on overexpression of TNFR2, and obviously the Miami Project for support of our lab and facilities and all the wonderful personnel. And also want to thank the other funding agencies and you all for remotely being there to listen to me for more than 55 minutes. And I want to thank you for your attention. I hope you're all safe. And if you have any questions, just feel free to ask. Thank you so much. Okay, so I can't hear. Okay, I see some questions that are here that I would like to address and try to address all of them. And if anyone wants to ask, they can open the microphone and let me know. I'll first address the first two. From Nagi, he is curious to understand why we didn't try the Rosa locus. If there any reason. The thing is that we were able to have this circular construct that allows the co-expression at the same time of TNFR2 and EGFP reporter. The fact that we had this concurrent activation using the TRE element under the control of the TRE element allows us to have a better time controlling and correspondence between the levels of TNFR2 protein in EGFP and back and forth. Most importantly, the EGFP works so well. It is important for us because TNFR2 antibodies really do not work well. I have other words for that. They're really a mess. And so EGFP reporter is pivotal for us, and in the system works. So we're not gonna use ROSA 26. I see a follow-up. As a dominant negative effect depending on the degree of expression and cell type signaling pathway action in cell type. Yes, if there is a way to look at these in sections, of course, the one way to assess it is also looking at what I was mentioning before, the NF-kappa-B signaling pathway, which I've done some staining for in slices before, and the antibodies work really well. So this is the main signaling cascade, which is downstream of TNFR1 and 2, and therefore these could be one of the way to address this. Now, Susanna was asking if there are pharmacological compounds that target TNFR2. Yes, there are, but the TNFR2 agonists that are known today, first of all, they're biologicals. So we do not have a specific chemical species. We have biologicals that do work. And there are some studies, very preliminary as well. But the main caveat with those is the fact that they do not pass the BBB, and therefore they have to be applied straight within the CNS, which is intracerebral, but also intrathecal, which renders the possible future applicability much harder. And one of the projects that we have in our lab is to also develop proper TNFR2 agonists. So yes, I think I've addressed all the questions that were addressed here. If there are any other, I'm open. All right, looks like.
Okay. It looks
Carlos,
like I think they've answered.
Carlos has a comment. Yes, please, Carlos. He can talk. Okay.
Okay, maybe now? Now I can, right?
Yes. Hi, Carlos. How are you?
Hi, how are you? That was a great talk. So I was, regarding Nagi's question, I was consulting with Roberta when she was doing this transgenic. And we went back and forth a lot regarding how to do the transgenic, because it could be random integration, as we did, she did, or put in the ROSA locus. The question, since it was an inducible model, and we looked around, we didn't see many examples of inducible transgenes into the ROSA locus that's supposed to be, you know, a safe harbor. So we discussed this a lot, and she finally decided, you know, just to go for random integration because of the lack of examples. Now maybe there are a few examples, but I'm still wondering if it's the best place to put or not. That's it.
Okay. Thank you so much for your comment, Carlos. As again, the data that I showed you, if I can comment on top of that, suggests that EGFP concurrent expression with the TNFR2 does not have any detrimental effect by itself. Something that definitely needs to be addressed, but whatever we saw in the female mice in the early dox paradigm, it is way more likely that is can be attributed to interference with the myelination process rather than the GFP expression by itself.